We had looked at tissue dynamics, and while we talked about tissue dynamics, we talked about what the cells would usually be doing when they are residing in the cell, residing in the tissue. And the first thing we talked about would be the cell division, so which is cells dividing and multiplying, and you actually have cell differentiation as the next step usually. Like if you have a stem cell, which is where the most of the tissues are going to originate from, and the cells would all come from stem cells, rather than mature cells multiplying to cause to create large number of cells. Usually for creating new tissues, stem cells are recruited, and you actually have stem cells multiplying to form the desired tissue. For that to happen, differentiation is a crucial factor. So we will talk about cell differentiation. So differentiation is basically the process of cells specializing in a function. So stem cells, as you all know, do not have any specialized function. They can mature to form any type of cell. So the differentiation is the process which results in the formation of a specific cell type with a desired functionality. So there can be cell differentiation because of the process called asymmetric division. Asymmetric cell division is basically where you have two non-identical cells which are formed. The daughter cells are not the same. So in regular mitosis, you would have two cells which are exactly the same. Here in asymmetric cell division, one is basically for the self renewal property of the stem cell. So it basically divides and retains its stemness. Other goes into the progenitor cell. And these progenitor cells can then become more and more differentiated to form the final differentiated cell lines. But usually these progenitor cells multiply through just regular cell division to form more number of progenitor cells, which then get differentiated. So this is because, as I already mentioned, progenitor cells divide at a much faster rate compared to fully differentiated cells. So what you are seeing in the figure is the first step is the asymmetric cell division, where the cells are dividing. One is forming a stem cell which is self renewing, the other one is a committed progenitor cell. The committed progenitor cell basically then multiplies and amplifies. This is the progenitor division, so that you have more number of cells that can be differentiated, and the final step is the terminal differentiation to form the fully differentiated cells. So during cell differentiation, you would experience something called population asymmetry. Basically divisions of stem cells population results in a large number of cells, and these can actually form different types of cells, and this provides additional versatility. Cells can proceed down several possible fates depending on the environment they are in, conditions that are provided to them, and so on. So if half the cells become stem cells, the total population of the stem cells remains a constant. So that way you have the self renewing property of the stem cells taken care of. So what actually happens in a differentiation process is, as a stem cell matures, it undergoes changes in its gene expression that limit the cell types that it can become. So there are changes inside the cell which make sure that it is beginning to get some of the functionalities of the differentiated cells, and it then, because this happens, it starts moving closer towards the final cell type. So these changes are actually monitored by can be monitored by the presence of proteins on the surface. So the surface markers and receptors which are expressed can be used as identifying whether the cell is remaining as a stem cell or if it is moving closer towards a differentiated cell. So with each successive change, the cell moves closer to the final differentiated cell, and its potential for becoming a different cell is actually becoming lesser. So it becomes more and more committed towards one particular cell type as more and more changes happen to the cell. So which is logical, right? So it is not a one step process. It is not like the stem cells form a committed progenitor which multiplies and then miraculously it just changes to one particular cell type. It does not happen that way. It is a sequential process which takes a significant amount of time, and as the time goes on, it gets more and more committed towards particular cell type. So this is an example of how it could actually happen. So what you see here is a totipotent cell which is non self renewing, like zygote. So, zygote does not self renew, right, and then you have the pluripotent stem cells which are self renewing, and embryonic stem cells would be an example of the pluripotent stem cells. So, then this forms a broad potential self renewing cell. So, these are still self renewing cells, these are multipotent stem cells which come from here, and then it becomes a limited potential limited self renewal cell, which until this process what you would see is there is a possibility of the cells going back, right. So, it can actually go back a stage because it is not fully committed yet. So, it can still go back, and here it forms some kind of a progenitor. So, here the example given is for neural cells. So, it forms a neural progenitor, and here it is more committed towards one particular lineage. So, from here it then divides where it is even more specific, where it can become a neuronal progenitor or a glial progenitor, and here by this point it becomes almost irreversible. So, after this it is now committed to a particular lineage and it cannot actually go back. So, and then finally it forms a neuron or a glia based on how the conditions are. The cells can probably die at any of these stages, but they cannot go back after it commits itself to a particular lineage. Okay, so, this would be how a general process would look like. So, I will talk about a couple of things which are very popular when we discuss stem cells and cell differentiation. One process which is very well studied and has been very well established is the hematopoietic stem cell, the process of hematopoiesis. The process of hematopoiesis is basically the process where hematopoietic stem cells commit and differentiate to form different blood cells, right. So, that is basically the formation of all the types of blood cells is from a type of cell called hematopoietic stem cell. So, these actually reside inside the bone marrow, and they have the ability to give rise to various types of mature blood cells and tissues. So, these are self renewing cells where the daughter cells can either be a hematopoietic stem cell or a myeloid or lymphoid progenitor cell. So, once it commits towards a myeloid or lymphoid progenitor, it will then go through the differentiation pathway and to form different types of cells. Okay. So, but it goes through asymmetric cell division first, so that the self renewing property of the hematopoietic stem cells is taken care of. The progenitor cells can follow any differentiation pathway. So, these myeloid or lymphoid progenitor cells are not going to form one type of cell, right. So, myeloid will have its own variety of cells and lymphoid can have its own variety of cells that can be formed. So, they can follow any of these pathways leading to production of one or more cell types, but these cell types cannot renew themselves. So, these progenitor cells cannot renew themselves, they are already committed. So, this is a simplistic representation of what hematopoiesis is. So, what you see here is the multipotential hematopoietic stem cell which has the self renewing property, then differentiating, multiplying to basically form a common myeloid progenitor or a common lymphoid progenitor. So, this is formed through asymmetric division. So, you either have another HSC formed or a common myeloid progenitor or a common lymphoid progenitor which is formed. So, from here, from the myeloid progenitor you can get megakaryocytes which form thrombocytes, erythrocytes, mast cells and myoblasts. Myoblasts can then go to form basophils, neutrophils, eosinophils, monocytes and macrophages. Common lymphoid progenitor cells can actually form the natural killer cells, or it could form the small lymphocyte which would then further be differentiated to form T cells and B cells. Right. So, this is a very simplistic representation of what a hematopoiesis process is. So, in general blood cells are actually divided into three lineages. You have the red blood cells or the erythrocytes, these are the oxygen carrying cells, they have the hemoglobin. So, they bind to oxygen and they can carry oxygen to the site. So, these are functional cells that are released into the blood. So, once the differentiation happens it will get released into the blood. They are formed by the process called erythropoiesis. So, within hematopoiesis there the pathway which is taken for differentiation of hematopoietic stem cells to form erythrocytes is called erythropoiesis. You also have lymphocytes which have a critical role in immune response and especially in adaptive immune systems. So, these are composed of the T cells, B cells and the NK cells or the natural killer cells. So, these are formed by the process of lymphopoiesis, and cells from a myeloid lineage include granulocytes, megakaryocytes and macrophages. So, these are derived from a common myeloid progenitor through myelopoiesis. This is also, these cells are also involved in innate immunity and blood clotting and many other mechanisms. They have a versatile functionality in your body. So, these are the three major lineages for blood cells. So, the process of forming granulocytes is called as granulopoiesis or granulocytopoiesis, and this is done inside the bone marrow except for mast cells, where the mast cells are actually produced. The differentiation happens outside the bone marrow, which is called this extramedullary maturation. So, medulla is the technical term for the bone marrow. So, it is extramedullary maturation. So, megakaryocytopoiesis is the hematopoiesis of megakaryocytes. So, this is the general hematopoiesis process. So, if you were to look at it in a more detailed step by step fashion, this is what it would look like. So, the multipotential hematopoietic stem cells have the self renewing property and they form common myeloid progenitors or common lymphoid progenitors, as I had already mentioned. And so, the common myeloid progenitor can basically come out of the bone marrow and then mature into a mast cell, that is the extramedullary maturation to form mast cells. So, inside the bone marrow, this myeloid progenitor can then become a megakaryoblast, which then becomes a promegakaryocyte and a megakaryocyte. So, this megakaryocyte then comes to the blood where it forms thrombocytes or platelets. So, this process is the thrombopoiesis, where you end up forming the platelets. So, the formation of megakaryocyte is megakaryocytopoiesis. Okay. So, the other pathway for erythropoiesis would be, it goes into the to form proerythroblast, basophilic erythroblast and then polychromatic erythroblast, orthochromatic erythroblast or a normoblast, and from where it forms a reticulocyte or a polychromatic erythrocyte. So, this enters into the bloodstream where in the form of erythrocytes where it gets matured. So, this pathway is the erythropoiesis. So, as you see, these are all coming into the blood, whereas mast cells enter into the tissue. So, from the common myeloid progenitor, you could also have the myeloblast, which can then differentiate into different granulocytes like basophils, neutrophils, eosinophils, and it can also form monocytes, which can then differentiate to form dendritic cells and macrophages. So, as you see, there are steps for each of them. So, it all goes through the promyelocyte, then to myelocyte, then metamyelocyte, then a band cell, finally forming either a basophil or a neutrophil or an eosinophil and so on. Okay. And so, what you need to notice here is where these differentiations happen. Right. So, until the last step, everything happens inside the bone marrow, and the final step is where it happens in the blood. The final maturation happens in the blood or it gets released into the blood. Okay. So, the common lymphoid progenitor, however, forms these lymphocytes, which goes through either a lymphoblast, prolymphocyte, and then forms small lymphocytes, which would then develop into B cells and T cells, or it could go into forming natural killer cells, which are the larger granular lymphocytes. And you could also have this common lymphoid progenitor forming dendritic cells, the lymphoid dendritic cells directly. Okay. So, as you see, there is a significant number of steps which are involved in each of these processes. It is not like, so unlike the previous thing which we saw. So, in the previous one, what we saw is it just goes from here to here, right. So, myeloid to erythrocyte is what a simple representation is, but in reality it actually has so many different steps. The cells exist in 4 or 5 different levels of differentiation before it finally forms the fully differentiated cell. Okay, so hematopoiesis can actually be modeled, and there are 2 theories for modeling it. One is the deterministic model and the other is a stochastic model. So, deterministic model is the classical way of describing hematopoiesis. What we kind of saw is a deterministic model where we say it goes into this environment will provide this particular condition where it goes into everything and it goes into a particular pathway, and there is no real randomness to this process. It just depends on the factors, the colony stimulating factors and other factors present in the microenvironment. So, this determines the path for the cell differentiation, right. So, this is what we kind of have always believed in, and this is a classical pathway. However, recent experiments are supporting the theory of stochastic differentiation. So, basically undifferentiated blood cells can differentiate into specific cell types by randomness. So, there is no real control over how this happens, and there are factors which do control, but there have been studies which show that the cells can go back and at each step, and there is quite a few there is a significant level of randomness which cannot be completely ignored. You cannot just say that it is deterministic, saying this environment will definitely lead to this. So, it is not that. So, so this actually is a representation of the growth factors and other molecules which can guide into specific differentiations. So, what you would see here is many of these factors are repetitive, which is probably one of the reasons for the stochastic model of differentiation, right. It is not that like for example, this IL-3 can be present in it is present in the formation of myeloid progenitor, which can then form megakaryocytes or erythrocytes. So, it is present in both. So, it could play a role in both these conditions, right. So, based on this, it is actually very difficult to say that one particular environment is going to force cells into dividing into forming particular things, but people are trying to use this understanding to design the cocktail in which the cells can be grown, so that there can be a differentiation that is directed towards a specific cell lineage. So, the next major type of stem cells which are extensively studied and extensively used in tissue engineering applications is the mesenchymal stem cells. So, these are basically multipotent stromal cells that can differentiate into a variety of cell types. So, basically they have been shown to form osteoblasts, chondrocytes, myocytes and adipocytes. So, osteoblasts are bone cells, chondrocytes are cartilage cells, myocytes are muscle cells and adipocytes are fat cells. So, mesenchymal stem cells have a very good capacity of self renewing while maintaining their multipotency. The capacity of the cells to proliferate and differentiate is known to decrease with the age of the donor as well as the time in culture. So, as even if it maintains its stemness, as you have multiple passages, then you would see that the rate of proliferation would be lesser and its ability to differentiate into different cell types comes down. So, the original source for mesenchymal stem cells is bone marrow. So, hematopoietic stem cells are also from bone marrow, right. However, these are different from the mesenchymal stem cells. The HSCs and MSCs are different, although they both reside in the bone marrow. Bone marrow mesenchymal stem cells do not contribute in the formation of blood cells, whereas hematopoietic stem cells are fully committed towards the formation of blood cells in your body. So, because they are not involved in the formation of blood cells, they do not express the HSC marker CD34. So, that is actually used to identify whether the cells you have isolated is a mesenchymal stem cell or a hematopoietic stem cell. So, these are also referred to as bone marrow stromal stem cells, and so these terms are used interchangeably in many cases. Another source for getting mesenchymal stem cells is the umbilical cord tissue. So, this is the youngest and the most primitive mesenchymal stem cells. That is why there is a lot of interest in storing the cord blood and the cord placenta, to hope that you would be able to use it for some regenerative medicine which might be developed in the future. So, this basically has two different tissues, one is the Wharton's jelly and the other is the cord blood. So, the Wharton's jelly is a gelatinous material which is present in your placenta, which is very rich in mesenchymal stem cells, and the cord blood is rich in hematopoietic stem cells. So, there are other sources as well. Adipose tissue is a rich source of adipose derived mesenchymal stem cells. Molar cells, which are basically the teeth tissue. So, if you have a developing tooth bud, then those are rich in mesenchymal stem cells. So, these mesenchymal stem cells or the dental stem cells actually form enamel, dentin, blood vessels, dental pulp and various nerve tissues. So, people have also shown that these can form hepatocytes and many other types of cells. Amniotic fluid is also a source from where you can get mesenchymal stem cells. So, it is the fluid in which the fetus is present, and this can actually be harvested using a process called amniocentesis. Amniocentesis is nothing but taking a large gauge needle and injecting it into the uterus to draw the amniotic fluid. So, this is usually done for diagnostic purposes. So, this amniotic fluid basically has a lot of cells, and 1 in 100 cells which are collected using amniocentesis is a mesenchymal stem cell, which could also be used for tissue engineering applications. So, this is a representation of how mesenchymal stem cells can actually differentiate to form various lineage cells. So, I am not going to go into great details. So, what I have here is for adipocytes, myocytes, chondrocytes and osteoblasts. So, within that we have basically classifications. We have the white adipocytes, brown adipocytes, skeletal muscle and cardiac muscle, smooth muscle, and chondrocytes and mature osteoblasts. Okay. So, this kind of gives what is the pathways, different pathways these mesenchymal stem cells can take, and what would be the growth factors and other molecules which can be helpful in directing the mesenchymal stem cells into these lineages. So, whatever we looked at, right. So, the basic concept of differentiation is what usually happens in your body. So, you want to try to use this to direct differentiation, ultimately that is the aim, right. We want to, because we are looking at a tissue engineering application where we are going to use these stem cells, and we would want to differentiate these stem cells to produce specific cells with desirable functionality, so that we will be able to perform the tissue we develop will be able to perform its functions. So, this is basically done through activating endogenous transcription factors, or it can also be done by transfection of stem cells with ubiquitously expressing transcription factors. You can also have the stem cells being exposed to selected growth factors, or you can culture the stem cells along with other cell types which can induce lineage of the specific cell type. So, you can also do combinations where you treat it with growth factors or antagonists and try to use this kind of a process for differentiating cells, directing the differentiation of stem cells. So, basically this is what happens. Let us look at an example where you are basically talking about an external signal, which is a growth factor which you add. So, let us say a growth factor is present in the media in which the cells are being cultured. What happens is this diffusible protein growth factors will bind to the receptor of the protein, will have to diffuse and reach the receptor of the surface of the protein, where it will bind to the receptor, and then it initiates an intracellular signal which is transmitted through protein tyrosine kinase or other intracellular signal transducers like G protein. And once that happens, there is an action within the nucleus which basically helps in which basically causes the expression of certain transcription factors, which results in the cells getting differentiated. Okay, so, this is what you see in an external signal stimuli. So, other than this, you can also direct differentiation using biophysical properties, which would be the mechanical or the physical properties of the material on which the cells are being cultured. So, this is a brief representation of what are all the factors that can actually affect. You could have material composition that could play a role. Depending on the material composition, how the cells interact with the material will change. If you the integrins and cell binding, cell adhesion could all change based on the composition of the material that you are using. Substrate stiffness has also been shown to affect how the cells differentiate themselves. And the last one is the nanotopography, where the surface topography of the material can actually direct cells, because that helps in while the cells are cultured, they might have to be aligned in the presence of this nanotopography, and if that happens, the cells will differentiate to specific cell types and so on. So, here are some examples where composition and stiffness and topography have been used to get different types of cells. So, I am not going to go into each and every one, but I hope you would go through this to try and understand what each of these materials and each of these properties can actually do. So, this gives an overview of what people have tried and what people have been able to accomplish. So, one last example would be alginate. Alginate is something which has been studied extensively for mesenchymal stem cell differentiation, because they are very easy to work with, and you can change their properties very easily. So, people have shown that the structure of the alginate, whether it is an injectable solution or if you have it as a porous scaffold, can alter the way these molecules are delivered, thereby that can play a role in how the mesenchymal stem cells are differentiating. Another factor which has been shown to modulate differentiation is the adhesiveness of the surface, the adhesivity of the alginate material. So, in the presence of a peptide, people have observed it to be differentiated to osteogenic, whereas when there is no peptide, it differentiates towards a chondrogenic cell line, even when every other property is the same. And composition can actually mediate the material properties, and it can be used to convert cells differentiate cells to form myogenic or chondrogenic, depending on whether you have collagen along with this or fibrin along with this. So, people have shown these kinds of differentiations are also possible. Mechanical properties such as stiffness, as the stiffness is lesser, you would end up forming something like an adipocyte, and as the stiffness increases, you would end up forming a muscle or a bone tissue. So, it is quite logical, right? It depends on how the ECM of the natural tissue is, and based on that this differentiation happens. Okay. So, this gives a brief introduction on the biology of differentiation and how directed differentiation is being explored for stem cells. So, in the next lecture we will talk about a little bit about the mathematical modeling aspects with respect to how these external factors interact with the surfaces and so on. Thank you.